Hello, welcome everybody. Today we are going to discuss a new topic about placenta and placental hormones. So at first we should know how placenta develops after the fertilization and implantation is complete. So in humans, the fertilization occurs, we know, at the ampulla of the fallopian tube. Now the fertilization process involves, number one, chemoattraction of the sperm to the ovum by the substances provided or produced by the ovum, number one. Number two, the sperm adheres to the zona pellucida, which is the membranous structure surrounding the ovum. Number three, the penetration of the zona pellucida and the acrosome reaction. And number four, the adherence of the sperm head to the cell membrane of the ovum and breakdown of the area of the fusion and releasing of the sperm nucleus into the ovum cytoplasm. Now, though the millions of the sperm are deposited in the vaginal tract, only 50 to 100 reach to the ovum and many of them contact with the zona pellucida. The sperm bind with a receptor in the zona pellucida and there is acrosomal reaction, which we already talked about in the point number 3, that is a penetration of the zona pellucida and the acrosome reaction. So what is the acrosomal reaction? It is nothing but the breakdown of the acrosome cap of the sperm, which is the lysosome-like organelle on the sperm head. So various enzymes are released, including the trypsin-like protease, acrosin, but it facilitates not only it's always required the sperm penetration in the zona pellucida, and the sperm reaches the ovum membrane and the fusion then is facilitated by the protein fertilin. It is a protein on the sperm head which resembles the viral fusion proteins which permits some viruses to attack cells. That means this fusion of the ovum membrane and the sperm is enriched by the protein fertilin, and this is a fusion protein situated on the sperm head, and it is nothing but resembling like a viral fusion protein. Like when virus attacks our cells, these viruses are attached with a host cell membrane by some fusion proteins, and this has some resemblance like this protein fertilin. Now this fusion, that means the fusion between the sperm and the ovum membrane, provides a signal which initiates the development and decreases the resting membrane potential of the ovum and thus inhibits the process of polyspermy. Polyspermy means only one sperm can penetrate one single ovum. Not more than one sperm can be allowed to penetrate a single ovum. This means it inhibits the polyspermy. So it follows by structural changes in the zona pellucida which also inhibit the polyspermy on a long-term basis. So after that, blastocysts moves down to uterus. The journey takes 3 days, during which it reaches 8 or 16 cell stage phase. After that, in the endometrium of the uterus, blastocyst is surrounded by an outer layer called as syncytotrophoblast. It's a multinucleated mass with no discernible cell boundaries. That means it has no definite type of cell boundary, but it's a multinuclear cell mass, and the inner layer of this cytotrophoblast, which is made up of the individual cells. So the syncytotrophoblast erodes the endometrium and the blastocyst burrows it into the endometrium. This process is called the implantation, and it is a site usually on the dorsal wall of the uterus, and from there placenta develops and the trophoblast remains associated with it. Next point, which is a very important point regarding this fact, that always we know a fetal is like a foreign body to a mother's womb or in mother's uterus. So in case of every foreign body, we know the foreign body is rejected by some immunological processes occurring in our body. But why the fetus or the fetal graft is not rejected, or it is failured to be rejected by the uterus? So let's take a look where there is a failure to reject the fetal graft by the uterus. So at first we should make this point very much clear. The mechanism of action of this process is still unknown. One explanation is the placental trophoblast, which separates the maternal and the fetal tissues, does not express the polymorphic class 1 and class 2 major histocompatibility complex proteins or genes, and instead it expresses a HLA-G protein, which is a non-polymorphic gene. Okay, so thus antibodies to the fetal tissues never develop. So whenever there is no antibody produced to some antigen, immunological process doesn't take place and there is no question about the rejection of the fetal graft. So also there is a second point, there is a FAS ligand on the surface of the placenta and it binds to the T cells, causing them to undergo apoptosis. So this protein helps the T cells to undergo apoptosis, and it's another cause which inhibit the rejection of the fetal graft by the mother's body.